far though, isn't it? For me at least.
Yeah, I think probably for you lot it's okay for me.
Right.
Yeah, but hopefully things will start to get back to normal. I think there's a date of the 12th of April. I think the government has allowed for outdoor activities to happen again. So hopefully that will take place. I'm not sure about large venues. My son is hoping to go to the Reading Festival, but I'm not sure if that's going to happen. Maybe a bit too large. Hi, Nicholas. How are you? Are we all here now?
Good. How are you, miss?
Yeah, good. Thank you, Nicholas. Very good. So let's see if we can take the register. I've got two computers, but unfortunately one is a lot slower than the other and it does actually freeze. So I might again have to take a paper registration. So let's have a look. We've got Abdina. Good morning. Oh, let's start with the registration. Michael, are you here? No. Chanel, good morning. Hi, Chanel. Can you just give me electronic wave, please? Thank you. Thanks, Chanel. Okay, Cedric, are you here? No. Okay, Rachel, good morning. Thanks, Rachel. Sanya, good morning. Thanks, Sanya. Okay, good morning, Lily. Is Lily not here? Okay, no. Kerwin, good morning, Kerwin.
Morning, miss.
Thanks, Kerwin. Good morning, Abdina.
Morning, miss.
Thank you very much, Abdina. Okay, Nicholas, good morning. Kylie, are you here? Kylie's not here.
Sorry, miss. Good morning.
Hi, Nicholas. Thank you. Good morning. Claudius, you're here. And Gianmarco. Thanks, Claudius. Is Gianmarco with us? Hi, good morning, Gianmarco. Thank you very much. Right, guys, we are continuing with the topic. It's actually quite a large topic that we are covering for AS level, okay? And today we are looking at mutations. Now, mutations is something that you've probably looked at in your GCSE year. Is that right? You've had probably a brief introduction to
Yes, miss.
mutations. Fantastic. So if I, sorry guys, I need to start record. Oh, it is recorded. So if I was to ask you to give me a definition on what is a mutation, could you have a little think and come up with a sentence? Just write something down now, and then I'm going to ask you to read out what your definition is, okay? So have a think, what is a mutation? And just put it into a sentence for me.
Miss, are you going to skip protein synthesis?
Am I going to skip which one?
Protein synthesis.
No, this just I'm doing it in order of the see, I've got all the PowerPoints on in order on AS level. So this is the AS level. So they're coming in sequence. Okay, so I can read them out to you. Okay.
That's fine. As long as we do it, I honestly don't mind what order you want to do it. It's
right. Yeah. Okay. So I'm just actually because I'm actually going in order of the PowerPoints because I should imagine they would actually are in a sequence for a certain reason. Okay.
Yeah, that's understandable.
Let me just tell you what they are. Bear with me, guys. Okay, so in terms of I just want to make sure. Bear with me. So I will tell you the order in which we are going for this. So 3.4, we have chromosomes, the triplet code, RNA, gene mutations, mitosis and meiosis, so that would be the next one. Non-disjunction, genetic diversity, types of selections, the concept of species, classification and taxonomy, investigation, diversity and evolution, again, more biodiversity and investigations of variations. So that, in fact, is pretty much what we're actually doing for AS level. Okay, so I'm going to, the mitosis and meiosis will be the next lesson, guys, okay? Right.
Yeah, that's fine, miss. Thank you.
So who would like to read out their definition for mutation? What is a mutation?
So I wrote, mutation is an error during transcription of a particular gene, which causes the gene to code for the particular characteristic differently in a way that may or may not offer an advantage to the organism.
Okay. Yep. Thank you, Nicholas. Okay, that's a really long definition there and quite detailed. Thank you very much. Abdullah, what did you write down for your definition?
Well, what I did mutations, I went off with the definition of, I think it was when we did HIV. That's what I went off the definition.
Okay, that's good. That's fine.
It wasn't. I think it was influenza. I think that could mutate, right? Am I correct?
Yeah.
Yeah. So like it can change. So what I would have mutated of like a pathogen like changing its like surface in proteins in order to look different.
Yeah. So mutations can occur through because of a toxin, and a virus would be one of them. Okay, so it can actually change the base of your DNA because of that. Absolutely. Thank you, Abdullah. Jamarco, what did you write? Oh, Jamarco, is that because your mic isn't working? Okay, fair enough, don't worry. Any of the ladies? Let's have some girls. Anybody would like to contribute? How about you, Cohen? What did you write?
A mutation can be described as a change in DNA / of a gene, and this can be due to mistakes during cell division or infections, for example.
Okay, lovely. Yeah, well done, Cohen. Thank you very much. That's really good. So you all understand the concept of mutations and where mutations occur. So fantastic. So gene mutation are changes to the base sequence or the quantity of DNA within a gene or section of a DNA. So I don't know if you want to write that down as well, just an addition of what you've also wrote, okay? I'll just repeat that. Gene mutations are changes to the base sequence or quantity of DNA within a gene or section of DNA.
One more time, miss?
Yep. So gene mutations are changes to the base sequence or quantity of DNA within a gene or section of DNA.
Or quantity of what?
Of DNA within a gene or section of DNA. And
Thickness.
That, in effect, definition should actually determine that there are mutations that are serious and also are known as large-scale mutations, and also mutations that are smaller mutations known as small-scale mutations. And we're going to have a look at the differences between the small and large scale mutations, okay, and what the definitions of them are. Now, I know, guys, sorry, my cat keeps coming in and out and keeps opening the door, which makes, I'm going to put the chair there. It just makes, because I've got different, it's very noisy outside this room, and he keeps coming in and out of this room, opening the door. Anyway, sorry. So I know, and I just want to do, so before we move on, okay, you pretty much know the difference between a gene and an allele. But can anybody please define what is a gene and what is an allele? Nicholas, I know that you know, but I just want to give somebody else another chance. Okay, thanks. I'll come back to you in a minute, Nicholas, if you don't mind. Hold your thought. Okay, I will ask you again, but can somebody please define? Go on, Cohen.
A gene is like, is it like part of a DNA that determines a trait, but then like an allele is like, it's a type of gene.
Yeah, you're on the definitely. Thank you, Cohen. So you are right, absolutely. So in terms of having to define or give an example, maybe to help your definition.
Okay. You have certain alleles for like different eye colors.
Yeah.
And then I'm not sure, for example. Like,
yeah, no, that's a very good example, Cohen. Okay, so all species, okay, so human beings, for example, will have the gene to code for eyes. Okay, your allele will give you is the characteristics of that eye. Will give you the eye color. Okay, so that in effect is pretty much all genes. So all species will have the same gene. It is your alleles that will actually give you the characteristics of those genes, if that makes sense. Nicholas, what were you going to say?
Yeah, basically what Cohen said. A gene is a subunit of DNA that codes for a particular trait. So you have the OCA2 gene, which codes for eye color. Like you said, you could have different alleles of this gene. So basically, different variants.
Yeah.
So I think for blue eyes, it's actually a mutation in that gene that causes the blue eye color, the blue phenotype. And you have dominant and recessive alleles. So if your mom has blue eyes but your dad has brown eyes, then I think you have at least a 75% chance of having what is it? At least a 75% chance of having brown eyes, depending on whether the actually the genotype of the parents, basically.
Yeah, absolutely. So your phenotype is what you display. Your genotype is also what you carry. So these chromosomes are carried from the gametes, 23 from the mother, 23 from the father. Okay. When a embryo takes place, so it is during the fertilization that these 23 chromosomes from each gamete then fuse, forming 46 chromosomes. And this is the process of meiosis, okay, where then the chromosomes, if in effect, start to cross over, and combinations of these alleles, if you like, in your genes, then create a new life. Okay, so this is combinations of what you would get from your mother and what you'd get from your father. We have one set of eyes, one pair of eyes, okay? So it might be that your genotype would be brown eyes, okay? But you've also carried that gene, that allele, from your other parent and passed that down, okay? So one you would display being your phenotype, and your genotype is also in your genes, but it's not what you would display, okay? So, and in effect, you would need 2 recessive genes for blue eyes, because blue eyes is recessive, and you only need 1 dominant gene to display that. So if you've got a capital B and a small b, capital B being dominant brown eyes, and lowercase b being blue eyes, okay, you've got a capital B, small b, you would actually display brown eyes being the dominant gene. You would need 2 recessive genes to display blue eyes. Okay, so let's move on now. So well done, you've got the concept and know the difference between a gene and an allele. Okay, and what we're going to have a look is the differences also between your small scale gene mutations and your large scale mutations. Now, your small scale mutations often are gene mutations. Okay, so these are usually within a base. Okay, so it could be one combination that's misaligned, okay, or missed. Whereas your large scale, it's usually within the chromosome. So you have sections where have been deleted or sections inserted, which then contributes to every amino acid that's processed after that mutation has changed, causing a frame shift effect. Okay, and this usually happens during meiosis. So the difference between mitosis and meiosis. Okay, with meiosis, this is when we're talking about our gametes. Mitosis is when the cells are replicating themselves before cell death. Okay, so let's move on. I hope that I hope you've understood most of that. Just a recap really from your GCSEs. Let's have a look here. So here we have our DNA, we've got our RNA, and we are coding for these proteins here at the end. So any changes to the
So you're on slide 2.
Moving on to slide 3 now.
Slide 3. Okay.
Yeah, so here we got the definition of mutations. We've already done our definition, guys, okay? So we did that earlier, we wrote our own definition. So any changes to the quantity or the base sequence of a DNA of an organism, okay? So we've established that. Let's move on to our cell, slide 4, okay? So mutations can occur in our gametes, okay? And somatic cells. What do we mean by somatic cells?
Is it non-sex cells?
Yeah, so your body cells. Yeah, absolutely. So write that down. If you're not familiar with the word somatic, can you just please define it as the non-gametes? So these are all the body cells, okay, apart from the gametes. Okay, so mutations can occur both within the gametes and other cells as well. Okay, the chances of mutations, and that's a very large number. Okay, usually about 1 in 100 million. Okay, so that's the chances of mutations. And faulty DNA can be repaired by specific enzymes. So if a mutation does occur, we also try to repair it as well. Okay, so if there's a misalignment that's taken place, our body will actually try and recognize that and try to correct it as well. Okay, so when we're talking about mutations that occur in gametes, these are usually inherited mutations. So if there has been a genetic disease within the family, for example, hemophiliacs, okay, in the father's side, that sperm cell could possibly carry that genetic condition. Okay, so
what's hemophilia?
It's where it's internal bleeding. Okay, so it's where, I mean, I think the royal family actually have that. It's where you haven't got any of the blood clotting factors. So if you do cut yourself, okay, you can actually bleed, and it's dangerous bleeding internally as well. So your body has these blood clotting factors to actually repair any openings, and hemophiliacs, they are missing this factor. Okay.
Okay, thank you.
Okay, I do think that the royal family actually have that genetic disease. I think not so much now, because where they used to marry, only royal blood could marry royal blood, and so now that they've actually
they used to marry into themselves.
Yeah,
families,
yeah,
didn't they?
Yeah, they did.
So,
yeah, they're just probably inbred, so obviously that will pass on.
And so that actually does occur with a lot of families as well, with obviously religious beliefs and stuff like that. And what is actually doing, the gene pool is very small. And so the likelihood of that child being born with that disease is going to be higher, because probably both parents are carrying that disease. So opening up the royal family, allowing them to marry commoners, if you like, has in fact opened up their gene pool. So I should imagine the likelihood of them being born now with haemophilic is actually quite low. Now that they can marry anybody, apart from Catholics. You still can't marry Catholics. Or anyone, I think, first in line to the throne can't. Anyway, that's something different. History is so interesting, isn't it? Anyway, let's move on, guys. Where have I got to? I've digressed. Oh, so small scale and large scale gene mutations. So when we're talking about our gametes, okay, so this would be, would you say that would be large or small scale mutation? If it is so when you're talking about our small scale gene mutations, okay, these are just usually substitutions that have taken place, and we're going to have a look at our bases, okay? My goodness, my phone keeps that's Mr. Hawkins. Let me just see what he wants. Sorry, guys. Okay, right, I've muted him. Okay, so yeah, let's move on. Let's have a look at the different types of mutations. So we have the so can you please write down small scale and large scale? Small scale usually refers to gene mutation, whereas the large scale usually refers to chromosomal mutations, okay? And these is large sections within, and it could be the whole chromosome has actually been affected, okay? So this is pieces of deletions and insertions as well. And this usually occurs during the process of meiosis. So this is where we're talking about our gametes, okay? So large scale mutations tend to be large sections of the chromosomes that have been affected, and this process usually occurs during meiosis, okay? And also inherited through gametes as well. So when we're talking about meiosis, this is where the chromosomes are overlapping. We've got 23 chromosomes from the father, 23 chromosomes from the mother, and during fertilization, meiosis takes place where these chromosomes join together and we have these overlaps of our genes. And this is why we get different characteristics on the offspring, some from the mother, some from the father, okay? Large-scale mutations can occur during this process, and also through inherited diseases such as, haemophilics, for example. Yes, Abdina?
Can you repeat the small scale and large scale one more time, please?
Yeah, of course. So we have the small scale mutations, usually just a gene mutation, okay? It's just a smaller sections. And these are usually a substitute for coding for the wrong amino acid, okay? Often it can be corrected because, as we know, we have 20 amino acids, but we've got 60 types of base pairing to actually make that particular amino acid. And so often it's not affected, okay? And so that's small scale.
So it's coding for the wrong amino acid, right?
Yeah, usually, but often it doesn't usually have an impact because we know that there are many combinations that actually make the same amino acid.
Okay, thank you.
Okay, and that would be a small scale. Our large scale mutation, okay, these are sections, larger sections of a chromosome, and it could be the whole chromosome that could be affected, and this is where deletions and insertions takes place when coding. And this means that every amino acid after the mutation has changed its whole frame, it's shifted, okay? So the whole combination after where the mutation has occurred can completely shift the sequence of the DNA of the amino acids produced after that.
Miss, which slide are you on?
Oh, I'm not on a slide. I'm just sorry guys, I'm just describing smaller scale mutations. I hope I'm not going too fast. I'm hoping I'm explaining it.
No.
Is that okay?
Yes, it's okay.
So we were talking about gametes and somatic cells. Usually, these are large-scale mutations that take place in our gametes purely because these are inherited. Okay, these are inherited diseases that can be passed on. Whereas where mutations take place in somatic cells, these can be the smaller scale. However, in somatic cells, if mutations occur in somatic cells, it usually leads to cancer. Okay, so these would be toxins. So if you've come into contact with radiation, for example, certain chemicals, a virus, that can actually change how the cells are replicated. Remember, your cells during mitosis are replicated before cell death. And so if a mutation takes place within that cell, it is also copied with that mutation. And that's how cancers usually occur. Guys, can I just have an electronic hand? I know I'm talking quite fast and I will slow down. I just want to know that you've understood so far. I just want to give you, thanks guys.
But do you say the large scale was inherited?
Yes, usually the larger scale mutations is usually through meiosis,
Yeah.
okay, and inherited gametes. Okay. So the gametes, so be it from the mother or from the egg or from the sperm, can also carry that mutation. Okay, so that's one type of large scale. The other type of large scale usually happens during the process of meiosis, where the lap over and the exchange of genes take place, where the 23 gametes from the sperm and 23 gametes from the egg fuse and creating 46 chromosomes.
Correctly when they overlap, basically.
Yeah, yeah. During the process of meiosis. Okay, so at least we have established small scale, large scale. We understand the difference between the gametes and somatic cells, okay? And it's usually we are able to repair any fault, okay? So certainly in somatic mutations, we will try to repair that, okay? And just to, mutations usually occur randomly, round about 1 in 100,000. Or guys, you've got 2 to 30 times 10 to the power 7, okay? It's very large numbers anyway. So I'm going to move on to slide 5, guys. So if we're not able to repair the mutation, that will affect new proteins being synthesized. Remember that the whole point of these codons is to actually make proteins, amino acids. And so the sequence of amino acids will be affected, therefore affecting what's the outcome, affecting the type of gene that's being produced. Okay, so we've got changes in an individual gene due to miscopying. Okay, so these are the types of mutations that occur now. All right. If you can write them down, I'm sure you know them all through GCSEs. So it's the changes in an individual gene. Okay. And this is a miscopying of one or more of nucleotides. We have deletions or insertions of a nucleotide resulting in a frame shift. That can also be a small scale, but usually it is a large scale, okay? There's a whole combination. All the amino acids, all the codons then have shifted, creating a completely different sequence of amino acids.
Wait, Missy, you said the codons have shifted.
Yeah, so if we have a misalignment, so yeah, completely. So if we are misreading, okay, or if something has been deleted, then for example, if we've started at A, T, G, if T wasn't there, so if you look at the first four letters, we've got C, A, T, G, okay? If T was deleted, that would then become A, G, C. Does that make sense, Abdina?
Or it'll come AGC, so that the mutation is when it's deleted. So T was deleted.
If T was deleted, okay, that would then become AGC, and that codon would make maybe a completely different amino acid.
Okay.
Can you see that the codon for ATG is codon 1, which is tyrosine?
Yeah.
If it became AGC, that could be something, that could be a different codon for a completely different sequence. It could end up being leucine or phenylalanine or something completely different. Yes.
Yeah.
Okay. Guys, if I can just have a show of hands that you've understood that concept.
It's what I said. A DNA sequence has shifted.
Yeah, it can completely shift. Absolutely. Thanks, guys. Yeah.
Wait, Miss, can I ask you a question?
Yes, of course you can.
Why would they like just delete or shift, or like what? How would that?
But so it can actually be genetic, okay? Also, toxins can actually, cause problems in. So if a cell becomes damaged, and so that can also shift the sequence as well. And so your DNA is only going to copy as good as the last one. And if there has been a misalignment or there's been a deletion or something due to different chemicals in the body or being exposed to toxins or anything like that, radiation, it can also then completely change the sequence of the DNA and it will replicate its last one.
Oh.
Okay.
All right.
Can you repeat that one more time, please, or you just repeat it to Kylie?
Yeah, absolutely. So when, and this is how cancers can occur, okay? So if we've become exposed to certain toxins, radiation or chemical or even a viruses, can actually damage the cell. And can actually, so when your bases become damaged, your base pairs then are coded differently. So it only needs one codon to be damaged for it to completely shift. And so it can occur through genetics, randomly, or through exposure to toxins as well. So with radiation, guys, it usually breaks apart the DNA strands. So radiation will damage your cells. And I think when you think about Rosalind Franklin, she actually was an example, a scientist, a pioneer scientist that actually founded DNA. Okay, so she eventually died because she used herself and she actually tried to take images of her cells through x-rays, if you like, and she exposed herself to lots of radiation. She died as a result of trying to discover DNA. Okay, and as a result, what actually happens at a cellular level, this is all happening at a cellular level, the DNA starts to break down or break apart, and then you have these misalignments of your codons. Okay. Is everyone okay with that? Let's move on, guys.
Yep.
So here's an example. Okay, so we've got, what did they just delete? Can you see that? So A has been taken out of codon 2. Did you see that? Mm-hmm. So So now, oh, okay, perfect example. So now codon 2 has become alanine because it actually was removed. Okay, so here we've got substitution mutations as a result of a change in a base. Okay, I'm going to go back, guys. Have you got access to move this yourself? These slides up and backwards and forwards.
Yeah.
Okay, have a go now of looking how that works. Okay, so have a look at the sequence, the bases, the codons, and then just have a look to see how that changes the codons for different amino acids. So guys, just have a look yourselves. I'll give you a few minutes to understand that. Okay, have you all had a go at doing that, yeah? Can I just also, when we're talking about somatic cells, if radiation, if any toxins have damaged our cells, okay, they are then called mutagens. So where a mutation takes place, it is known as a mutagen. Can you please write that down? So mutagens can have an impact on the, so when we think about the number of mutations that occur randomly, 1 in 100,000, if you have been exposed to toxins, then those mutagens are increased. So if you think for every 100,000, one codon can actually be wrong, and our body will try to correct that, and it does usually successfully. However, if you are exposed to these toxins, those mutagens are the chances of those mutagens, or 1 in 100,000, is increased. Okay, and that then in fact can be carcinogenic. Carcinogenic means cancer. Okay.
It makes me say if it's exposed to toxins, it could produce mutons.
Mutagens. I'll write that down for you guys. So these are mutations. Okay, mutagens are the mutations, and we have increased numbers of them if you're exposed to toxins. Okay, more mutagens that take place, the greater the chances of carcinogens. Okay, and that carcinogens is cancerous cells.
Basically, like, more chance of what again? More chance of.
You have a greater chance of carcinogens. Now, write carcinogens down. Okay, again, it's a new word that means cancerous cells. Okay, or can cause cancerous cells. So carcinogens, for example, we are exposed to carcinogens every single day, and we have our antibodies actually prevents this from happening. Okay, so we have a defense system as well. You know, we have our antitoxins. This is why we're saying it's really important to make sure that you have a varied diet. It stops these free radicals, okay, preventing carcinogens. So eating burnt toast is a carcinogen. Okay, burnt food can actually unravel your cells, if you like. It promotes free radicals, and that's going into the chemistry side there. I just want to focus more on mutations here. So it's a fantastic topic, and that is a much higher level. So I just want to make sure that we are sticking to the AS level, okay? So let's move on. So here you've been able to have a look at what happens. You've moved up and down these slides, have you guys? Have you seen how the substitutes takes place and how the codons? Yeah, perfect. Well, okay, we've got gene deletion as well and duplication. So we have an extra. Fantastic, brilliant. Guys, that's pretty much our lesson for today. So here we can just, we've got an extra chromosome, okay? So we have an insertion of an extra chromosome, and that is carried through the female gamete. And that then is the number of chromosomes for any offspring born with Down syndrome. So the production of new superior proteins result, okay, the gain of a production is advantageous. If there's neutral, there's no change, okay, the production of inferior or no protein results in fatal or disease causing. So guys, that's a summary of what we've just learnt now. That's pretty much our lesson for today. I'm going to wrap this up now, guys, and I will actually put some worksheets for you on Sims. Sim student, isn't it? That's what you call it, Sim student, is that right? Or would you, so I'm going to have a look to see if I can find some worksheets to actually go with today's lesson on mutations, and then you can have a go yourself at seeing if you can actually answer some of those questions today. That's pretty much it really, and I will speak to you next week. I don't have you, do I have you tomorrow? Let me just double check. Do I, Abdina?
You have to go in for a COVID test tomorrow.
Oh, you got to come in. Okay.
Yeah, miss, we'll see you on Monday.
Yeah, all right then. So guys, what I'll do, I will put up some questions for you to worksheets on mutations. Have a little go yourselves, okay? And then we can have a look on Monday. So we can do that if you like, okay? So have a lovely weekend. I'm not going to see you now until Monday. So I'm looking forward to seeing you all in school on Monday. Have a lovely weekend. Make sure that you're rested and you're up and ready for school Monday morning. Okay, have a lovely weekend. Bye, guys.
Thank you, miss.
Thanks,
You
miss.
too.
You too.
Bye now. Bye-bye.
Bye.
Bye.
Miss. Okay, goodbye.